Okay, so I'm going to go over the cell membrane and how different substances can be transported across the cell membrane. So, the cell membrane is a very important component of both prokaryotic and eukaryotic cells. So, the cell membrane is kind of like a security guard. So, it regulates what goes in and what goes out of the cell based on its structure. So the structure of the cell membrane consists of two phospholipid layers, which is why it's called the phospholipid bilayer. So we have one layer of phospholipids here and then one layer of phospholipids here. So this makes up the entire cell membrane. So the cell membrane has one side that's filled with phospholipids and then on the other side filled with phospholipids. So the reason why it's called a phospholipid bilayer is because we have a phosphate group connected to a lipid. So if you recall, a lipid is composed of glycerol and fatty acids, and then the phosphate group is PO43 minus here. So a phosphate attached to a lipid makes it a phospholipid. So on our phospholipid itself, we have the hydrophilic head. So the head part of the phospholipid is hydrophilic, meaning that it is water loving. So water loving means that polar molecules like water are naturally going to be attracted to that end. So it's going to bind easily. There's no weird attractions. So the hydrophilic head is water loving. So what makes up the hydrophilic head are the glycerol and the phosphate groups. So the glycerol and the phosphate groups make up the hydrophilic head of the phospholipid. And then our tails, what makes up the inside of the cell membrane, are hydrophobic. So these are water fearing, meaning molecules that are nonpolar, typically not the same as water. So if you remember back in Unit 2 when we talked about how polar solutes dissolve in polar solvents. So in this time, hydrophobic means that the molecules in the hydrophobic tails must also be nonpolar. So nonpolar molecules are not water loving. They're not polar like water molecules. So the fatty acids make up our hydrophobic tails on our phospholipid. So other than the phospholipid bilayer, which is pictured here in red, and then the tails inside the cell membrane, we have many different macromolecules that are found embedded on our cell membrane. So first we have proteins. So all of these blue particles here are all of your proteins. So channel proteins are important for cell transport. These integral membrane proteins and peripheral membrane proteins are usually meant to just keep the structure of the phospholipid bilayer. Just like the fluid mosaic model, which talks about how proteins are embedded on the cell membrane, kind of like a mosaic. So, channel proteins are important for how different molecules can be transported across the cell membrane. The rest of the proteins are there to keep the structure of the cell membrane because if it weren't there, it's just one floppy mess. Next, we have carbohydrate chains. So if you notice, the carbohydrate chains are located on the outside of the cell membrane. So these right here are going to be attached to the hydrophilic ends of the cell membrane. So a couple of these ones here are consisted of both a protein and a carbohydrate. So I believe they're called glycoprotein technically, which is why they're pictured in blue here. So the green ones here are all carbohydrate chains. And then last one, inside the cell membrane. So within the cell membrane itself, we have what we call cholesterol. So cholesterol is a type of steroid, steroid is a type of lipid. So cholesterol keeps the structure of the cell membrane. And since it's located in the hydrophobic tails, we know that cholesterol is also a nonpolar molecule because it is attracted to the hydrophobic regions of our phospholipid bilayer. All right. So that's the structure of our cell membrane. So make sure you know the structure of the cell membrane and which parts are hydrophilic and which parts are hydrophobic, as well as which macromolecules make up which parts of the cell membrane. All right, moving on to cell transport. So just to review before we move on, a solute versus a solvent. So a solvent is what you're dissolving your solute in. Our solute are going to be particles that are going to be dissolved in the solvent. So we're going to use the term solute and solvent a lot. Water is going to be the solvent. Water is almost always a solvent because it's a universal solvent. And then different solutes, oops, are like sugar molecules, salt molecules, etc. All right, so passive transport. The idea of passive transport is that solutes will move from an area of high concentration to an area of low concentration. So, what that means, if we look at this picture here, you can see that there are about 15 different solute particles. So, this side of the cell, well, that's the outer, sorry. So the outside of the cell has a higher concentration of solute compared to the inside of the cell. So we have a higher concentration of solute on the outside compared to the inside. So the idea of passive transport is that we want to reach equilibrium. We want to reach a point where it's going to be equal. So these solute particles are going to want to move in order to maintain equilibrium and to keep the concentrations equal inside and outside of the cell. So now we have solute particles moving into the cell because it's trying to move from an area of high concentration to low concentration. So that's called moving down the concentration gradient, and that does not require energy from the cell. So think of it as a room of 50 students versus next door in a room of two students. Students are going to want to move from the 50 student room to the two student room, ideally to a point where both sides will have 26 students. So ideally, once equilibrium is reached, um, they're going to go back and forth equally to maintain that same concentration. So when we have high concentration versus low concentration, you're going to see particles go across the cell membrane in order to go from an area of high concentration to low concentration through passive transport. And they want to do that in order to maintain equilibrium. And keep in mind, this is an easy process. It does not require energy from the cell. So, diffusion is a type of passive transport. So, diffusion is when we see different particles move from a higher concentration, an area of higher concentration to an area of lower concentration. So once again, it's called moving down its concentration gradient. So keep in mind, this whole big idea is passive transport, not requiring energy from the cell, and diffusion is the movement of solute particles across the membrane from high to low concentration. So keep in mind that in order to diffuse across the cell membrane, only small nonpolar molecules can do this without the help of any of the membrane proteins. And the reason why is because some particles cannot move across the hydrophobic regions. So small nonpolar molecules like oxygen and carbon dioxide can easily move through the hydrophobic tails without any help. So for simple diffusion, diffusing across a cell membrane without any help from any of the membrane proteins, it must be a small nonpolar molecule. And here are some examples: oxygen gas and carbon dioxide. So here is a simple diagram of simple diffusion. We're moving from an area of high concentration to an area of low concentration, as indicated by the arrow here. So ideally, until both sides are relatively equal. So until equilibrium is met. Facilitated diffusion. So this is another type of passive transport. In facilitated diffusion, we have protein channels or carrier proteins that help molecules go through the membrane. Once again, it's diffusion, so we're still moving from an area of high concentration to an area of low concentration. So protein channels are going to be used for ions and even different polar molecules. So these are called protein channels. They're the ones that looks like a slide. And then naturally, carrier proteins are used for larger molecules. So same thing, we're still diffusing across the membrane, going from an area of high concentration to low concentration. But this time, because they can't easily go through the non-polar hydrophobic tails region, it will need to travel through the protein channels embedded in the membrane. So polar molecules, they can't go through the hydrophobic tails. Ions are also polar, so they cannot go through the hydrophobic tails. Carrier proteins are used for larger molecules like sucrose, which are not going to fit in the channel proteins. So carrier proteins will change shape in order to fit the particle through. And the last type of passive transport is osmosis. So the idea of osmosis is specifically the movement of solvent particles only. So water is a solvent. So osmosis refers to the movement of water from an area of high concentration to low concentration. So if you take a look at this picture here, we have two sides of a tube, and there is a semi-permeable layer in between. So here we see a lower concentration of sugar molecules seen in green. And here we have a higher concentration of sugar molecules seen in green. So this membrane is semi-permeable, meaning only some substances can go through and not all substances can go through. So if you look at here in this membrane, we have only channels. They're specifically called aquaporins that allow water molecules to go through. So in order to make sure that the concentration of both sides are equal without the sugar molecules moving, water is going to travel to the other side through osmosis. So if we add more water on here, this will be less concentrated, and this side will be equally as concentrated as this side. So if you were to take like this area versus this area, the type of pressure that each sugar molecule is feeling is going to be relatively equal. So here we have solute particles that cannot move across a semi-permeable membrane. So water molecules are going to move across in order to equalize that. So once again, osmosis is only the diffusion of specifically water molecules, and the diffusion of water molecules is used to keep the solute concentrations equal on both sides of the membrane. And then the specific protein channels are called aquaporins. So I do have you write this down in your notes. These are specialized protein channels that only allow water molecules to go through. So steroid here, it's nonpolar, oxygen, nonpolar, carbon dioxide, nonpolar. Water here, however, needs to travel through aquaporins because it is a polar molecule and it can't easily diffuse across the hydrophobic tails. Right. So once again, here's another picture that indicates that. We can see that water is going to move from here to here in order to equalize the concentration of solute on both sides of the membrane. So since the membrane is not permeable to certain solutes, we have water moving across so that equilibrium can still be met. So once again, the idea of water movement is net movement. So water's always moving in and out, but the net movement of water is ultimately going to go from the area of the so it's a little flipped, but the net movement of water is going from an area of low solute concentration to an area of high solute concentration. So that's a little confusing. But if you look at it this way, we can see how water is used to dilute the concentration so that they're equal on both sides. All right. And then we have different types of solution. So when our solute concentration outside the cell is higher, but solute can't move inside the cell, water is going to move out in order to make sure that it's equal on both sides. So when that cell is in a situation where water is moving outside of the cell in order to equalize the concentration, we call that a hypertonic solution. So when water moves out of the cell, that's called hypertonic. And that happens when the outside concentration of solute is higher than the inside concentration of solute. Isotonic is when our concentrations of solute are equal both inside and outside of the cell. So in this case, there's not going to be any net movement of water molecules in or out of the cell. So in this case, if water's not moving in or out of the cell is going to stay the same shape. This case, if water is moving out of the cell, you're going to see the cell start to shrivel up and also to shrink in size. And then last one, if we have a hypotonic solution, our cell is placed in an environment where the solute concentration is higher inside the cell than it is outside. So what happens here is water needs to go in order to balance the concentrations of solute inside and outside the cell. So when that happens, water moves in, your cell is going to increase in size. So how this looks in an animal cell, it's normal for it to be an isotonic solution where water is in and out, so there's no net movement of water anywhere. If it is a hypertonic solution, water moves out, it's going to shrivel up. And then if water's moving in, it's going to burst. So that's what we call lysed. Versus in a plant cell, because of the presence of a cell wall, if we have water moving out, it's going to become shriveled and plasmalized. So your central vacuole is going to shrink a lot in size. Flaccid is when you have no net movement of water in or out. So it's going to be relatively soft. And then turgid, which is the environment it prefers, we have net movement of water in. But if you look here, a plant cell will not lyse like an animal cell will because a plant cell has a cell wall to prevent lysis from occurring.